Hello, I'm Silvia Bowen, head of the autoimmunity section at the National Institute of Allergy and Infectious Diseases at NIH. I'm excited to present our recent data included in this JCI manuscript describing the role of inflammatory type 2 dendritic cells in end-stage lupus nephritis. Our group has a general interest in the way infections can alter the progression of autoimmune disease. In this particular case, we follow up on the observation that infection with a malaria parasite has a protective effect on lupus nephritis. And this has been supported over the years in both human and murine studies. We now provide a mechanistic explanation for this effect that argues for a kidney-specific protection by the parasite and that places the focus on infiltrated dendritic cells. Laura Amo, a very talented postdoctoral fellow in my group, has been the main force behind all these experiments, so I'll let her explain the details next.
Hello, my name is Laura Amo. I am working as a postdoctoral researcher here in Silvia Boland's laboratory, and I'm delighted to give you an overview of the fascinating work that I have done here at NIH. In this work, we explore the progression of lupus disease using a well-characterized mouse model of lupus, Fc-gamma-R2b knockout. These mice develop spontaneous lupus-like disease characterized by chronic systemic immune activation, high serum autoantibody titers, widespread vasculitis, and lethal lupus nephritis. The nephritic process begins with the deposition of autoantibodies in the kidney forming immunocomplexes. This induces local expression of inflammatory mediators, glomerular inflammation, and leukocyte infiltration, causing severe tissue injury that is lethal for these mice. In this project, we learned the essential role of CCL17 producing inflammatory type 2 dendritic cells in the transition to end-stage lupus nephritis. We uncovered the specific requirements from end-stage nephritis through the study of differential effects post-infection with a malaria parasite. We use a non-lethal malaria parasite that performs a self-resolved infection that lasts for 20 days. We infected wild-type and lupus mice at presymptomatic age, and we follow them for signs of lupus disease for up to 12 months. All lupus mice infected with the parasite survived past 9 months of age. Interestingly, we saw that Plasmodium infection prevented lethal nephritis specifically, while increasing systemic autoimmunity. Particularly, the infection did not alter the levels of systemic inflammatory mediators or explain inflammation in long term. The ANA test revealed that Plasmodium-infected wild-type and lupus mice had higher autoantibody titers than the controls. However, we detected a significant improvement in kidney function in lupus mice after the infection. Proteinuria levels were significantly lower than control mice in long term. To evaluate the specificity of this protection, we compared the status of the kidney and liver 5 months after infection. While malaria-infected mice showed a reduction of leukocyte infiltration in the kidney, the liver had similar levels of leukocyte accumulation regardless of the infection status. After extensive analysis of the local response in the kidney, we found that the infection does not alter the deposition of immunocomplexes, the expression of innate cytokines, or the glomerular inflammation. This suggests that the protection occurs beyond this stage of lupus nephritis. Indeed, we detected a reduction in leukocyte infiltrates and in tissue damage, measured by glomerular destruction and protein deposits, among others. Therefore, the results revealed that parasite infection prevents severe kidney damage, but not glomerular inflammation. Among all transcripts analyzed in the kidney, only CCL17 was significantly reduced in the kidney after infection in lupus and wild-type mice. This chemokine was expressed by kidney myeloid cells and correlated with kidney damage. In vivo blocking experiments revealed the important role of this chemokine in nephritis. Lupus mice receiving anti-CCL17 blocking antibody had a significant reduction of nephritis, measured by proteinuria and leukocyte infiltration. As was the case with Plasmodium-infected mice, CCL17 depletion protects kidney disease with no effect on systemic inflammation or circulating autoantibodies. Furthermore, we detected a significant reduction in CCL17 producing dendritic cells in the kidney of infected mice, cells that were correlated with severe nephritis in lupus control mice. We phenotypically characterized these cells as type 2 inflammatory dendritic cells. To assess whether plasmodium-induced alterations in bone marrow cells were the cause of the reduced kidney infiltrates, we reconstituted irradiated lupus mice with bone marrow cells from previously infected or uninfected lupus mice. Mice receiving previously infected bone marrow underlie the renal protective effect of a parasitic infection, which means a reduction of proteinuria and infiltrates without altering systemic autoimmunity. Finally, to assess whether the bone marrow alterations induced by the infection were specifically targeting kidney-infiltrated dendritic cells, we generated chimeras with a 1-to-1 mix of bone marrow from infected and uninfected donors. Analysis of kidney-infiltrating populations confirmed that mice reconstituted with infected bone marrow prevented leukocyte infiltrates, while those reconstituted with an infected bone marrow developed full pathology in the kidney. Interestingly, the results revealed that out of all the CD45 positive populations present in the kidney of reconstituted chimera, the dendritic cells were the ones primarily biased against those of CD45.1 allotype, which marks cells derived from infected bone marrow. These results suggest that Plasmodium infection induces long-term alterations in bone marrow cells that specifically hinders the ability of dendritic cells to infiltrate the kidney. So overall, from this work we learned not only that Plasmodium infection protects from lupus nephritis, but also that the infiltration of inflammatory type 2 dendritic cells into the kidneys is an essential regulated event in the transition from manageable glomerulonephritis to lethal tubular injury.